Every year, American gardeners pour over $50 billion into fertilizers, pesticides, and soil products that treat the surface of the problem and ignore what lives beneath it. Bags of nitrogen that vanish in the first rain. Fungicides that kill the very organisms feeding your roots. Meanwhile, 5 soil practices used by farmers for thousands of years can permanently multiply the living population of your soil, triple the productivity of dead ground, and cost almost nothing to implement. The agrochemical industry generates hundreds of billions of dollars annually by making sure you never connect these practices to the yields they produce. The record opens underground. 1 teaspoon of healthy soil contains more living organisms than there are humans on Earth. Bacteria, fungi, protozoa, nematodes, archaea, and microscopic arthropods all form a web so dense and so active that scientists now call it the most complex ecosystem on the planet. In the rhizosphere, the thin zone of soil surrounding plant roots, microbial populations explode to 100 times the density of surrounding bulk soil. Plants are not passive users of this network. They feed it deliberately, sending between 20 and 40% of every sugar they produce through photosynthesis directly down into their roots to feed the organisms that feed them back. So what happened to that knowledge? In 1909, a German chemist named Fritz Haber synthesized ammonia from atmospheric nitrogen. Within a generation, the living soil was buried under synthetic inputs that bypassed biology entirely. The microbes starved. The fungal networks collapsed. An industry was born, built on a single myth, that the soil is a sterile medium, and all nutrition must be purchased. But the soil was never sterile. It was alive. We simply stopped feeding it. A 2022 meta-analysis published in Science of the Total Environment, tracking dozens of controlled studies across multiple continents, confirmed that organic amendments and microbe-supporting practices directly increase microbial diversity, microbial functionality, and crop yields, with microbial diversity positively correlated to harvest output through enhanced nutrient cycling. A 2025 review in Discover Agriculture went further, documenting soil microbiomes as a verified pathway to higher yields, nutrient efficiency, and stress resistance across crop types. The knowledge was never lost. It was simply made inconvenient. Until now. Here are the 5 soil secrets they never connected for you. Secret 1. Feed the microbes with organic amendments. The bag of fertilizer in your garage tells you 3 numbers. It does not tell you what those numbers cannot do. Synthetic nitrogen feeds the plant directly for a few weeks, and then leaches away. Organic amendments, compost, aged manure, and crop residues feed something else entirely. They feed the living population that feeds your plants for every season that follows. The mechanism is elegant. Microbes decompose organic matter into plant-available nutrients on demand, governed by soil temperature, moisture, and root signaling, not by a calendar schedule. When your tomato plant needs nitrogen in early July, bacteria around its roots mineralize organic nitrogen from last fall's compost and deliver it within hours. When demand drops, microbial activity drops with it. Nothing is wasted. Nothing leaches into waterways. A 2022 meta-analysis by Xiangyang Xu and colleagues, published in Science of the Total Environment, found that organic amendments significantly increased the Shannon Diversity Index, microbial richness, and phylogenetic diversity in treated soils, while simultaneously increasing crop yields across grain, vegetable, and legume categories. A 2023 open access paper in PMC documented even stronger effects in upland soils, linking organic amendment applications directly to measurable gains in microbial biomass and plant productivity. A separate 2023 meta-analysis focused on soil organic carbon confirmed that compost is consistently the most effective single input for boosting active carbon fractions and microbial diversity together. The deeper principle is directional. Synthetic fertilizer systems degrade the biology over time, which requires higher doses each season to produce the same output. Compost and organic amendments work in the opposite direction. Each application raises the biological baseline for the next season. You build a soil that needs less over time, not more. Apply 2 to 3 inches of finished compost across your beds every fall. Layer aged manure, leaf mold, or chopped crop residues through the growing season as top dressing. Within 2 seasons, your microbial biomass can double or triple, and the harvest responds in kind. A depleted garden bed amended consistently with compost has shown yield increases of 30 to 50% in home-scale trials, and in severely depleted soils, the gains run higher still. Secret 2. Stop tilling and let the fungal networks rebuild. Every time a rototiller passes through soil, it shreds the mycorrhizal fungal networks that took months or years to build. Those networks are not optional infrastructure. They extend the effective reach of plant roots by a factor of 10 to 100, delivering phosphorus, water, and micronutrients to crops in exchange for root sugars. Tillage destroys them in minutes. A tilled garden must rebuild its biology from scratch every single spring. The long-term data is now unambiguous. A 30-plus-year study at Michigan State University's Kellogg Biological Station, published through Global Change Biology, found that no-till plots produced corn, soy, and wheat yields roughly 40 bushels per acre higher than conventionally tilled plots, year after year, with the gap widening as the soil biology deepened. The study documented a systemic shift in microbial community composition away from fast-growing opportunistic bacteria and toward slow-growing, carbon-efficient organisms that improve long-term soil structure. A 2024 field study tracking 18 years of continuous no-till management documented increased soil organic matter, higher total nitrogen, more available phosphorus, greater bacterial community complexity, and a soybean yield increase of 8.5% compared to conventionally tilled controls. Meta-analyses published between 2021 and 2025 confirmed that no-till benefits compound over time and are especially pronounced in drier climates, where the preserved fungal networks dramatically improve water retention and drought resilience. The method for a home garden is simple. Stop digging. Stop rototilling. Add compost and organic matter as a top layer each fall, and let earthworms and microbes incorporate it downward through the profile. Plant directly into the undisturbed surface. The first season, results may be modest. By the third season, the fungal networks have rebuilt, and the yield response becomes undeniable. Secret 3. Keep living roots in the ground year-round. This is the secret that almost nothing in the synthetic system can replicate. Living roots are the engine of the entire soil food web. Between 20 and 40% of every sugar a plant produces through photosynthesis is pumped directly into the rhizosphere as root exudates compounds specifically designed to feed beneficial microbes. When roots die, the feeding stops. When soil sits bare between crops, the microbial population collapses. Cover crops solve this. A strip of cereal rye, crimson clover, tillage radish, or hairy vetch grown in the off-season does more than prevent erosion. It pumps living carbon underground at exactly the moment your cash crop beds would otherwise be biologically empty. A 2025 Penn State Extension review synthesized decades of research and documented that cover crops increase microbial biomass, microbial diversity, and microbial activity, with diverse mixes outperforming single-species plantings. The review confirmed significant boosts to arbuscular mycorrhizal fungi populations, which can reduce phosphorus fertilizer requirements by up to 50%, and to nitrogen-fixing bacteria, which deliver free atmospheric nitrogen to following crops. A 5-year field study published in 2025 documented cover crop rotations increasing soybean yields by approximately 8%, while measurably shifting rhizosphere microbial communities toward beneficial groups including actinobacteria. The compounding effect is the critical point. Cover crops do not produce a harvest you eat. They produce the biology that makes every future harvest larger. In a home garden, a simple fall planting of winter rye and crimson clover, terminated in early spring by cutting at the soil line and left as a mulch, can raise the productivity of the following tomato or squash crop by 20 to 40%, while cutting fertilizer needs nearly in half. Secret 4. Mulch every square inch of exposed soil. Bare soil is a wound. Direct sunlight cooks the top inch to temperatures that kill microbes outright. Rain impact compacts the surface into a crust that cuts off oxygen to the biology below. Wind strips moisture and carries away the fine particles microbes need to colonize. A bare garden bed in midsummer can lose 60 to 80% of its active microbial population within a few weeks of exposure. Mulch reverses every part of that damage simultaneously. A 2 to 4 inch layer of straw, wood chips, shredded leaves, or chopped cover crop residue moderates soil temperature, retains moisture at root depth, prevents compaction, and provides a continuous, slow food supply to decomposer organisms. The microbes eat from the underside of the mulch layer upward, converting it gradually into humus that feeds the next generation of plants. A series of meta-analyses published between 2021 and 2026 confirmed that organic mulching significantly increases crop yields and measurably enhances microbial diversity and richness. One meta-analysis documented yield increases averaging 48.6% in mulched systems compared to bare soil controls. A 2026 study tracking different mulch types across multiple crop systems confirmed that organic mulches in particular produce profound and consistent shifts in microbial community composition toward beneficial decomposers and plant growth-promoting organisms. The practical application is straightforward. After every planting, cover every inch of exposed soil with organic mulch at a depth of at least 2 inches, and 3 to 4 inches is better. Replenish the mulch as the bottom layer decomposes into the soil. The mulch is not decoration. It is the roof of the microbial house. Leave it in place, and the house fills with life. Secret 5. Increase plant diversity above ground, and the microbiome explodes below. The final secret is the one that ties the other 4 together. Every plant species cultivates a slightly different community of microbes in its rhizosphere through the specific chemistry of its root exudates. A monoculture bed, one crop grown alone season after season, cultivates a narrow microbial community vulnerable to disease, pest pressure, and nutrient imbalance. A diverse bed where multiple species grow together or rotate through the season cultivates a microbial community that is broader, more resilient, and more functionally complete. A 2023 microcosm study published in a major soil science journal documented a correlation coefficient of 0.79 between soil microbial biodiversity and ecosystem multifunctionality, a statistical relationship so strong that it positions microbial diversity as one of the primary drivers of productivity in any soil system. A 2021 field study comparing polyculture plantings against monocultures found that diverse crop mixtures shifted microbial communities toward plant growth, promoting actinobacteria, and produced yield gains of 15 to 35%, even before accounting for the reduced pest and disease losses that diversity provides. A 2025 review in Discover Agriculture by Pandey and Saharan synthesized the global evidence and concluded that microbiome-friendly practices, including diversity, rotation, and polyculture, offer a direct pathway to higher yields, higher nutrient use efficiency, and stronger resistance to drought, heat, and disease, while simultaneously reducing synthetic input requirements across crop categories. The method for a home garden requires no special equipment. Rotate your plant families across beds every season and never grow the same crop in the same spot 2 years running. Interplant compatible species within a bed: basil with tomatoes, beans with corn, squash sprawling below sunflowers. Add a few strips of flowering herbs or perennials permanently into the garden to anchor a stable microbial baseline that the annual crops can draw from. The more different roots you put in the ground, the more different microbes colonize the soil, and the more complete the nutrient cycling becomes. But it survived. The knowledge of soil biology never disappeared into corporate archives or regulatory filings. While the synthetic input industry consolidated into a system worth hundreds of billions of dollars, a quiet network of researchers, indigenous farmers, cover crop advocates, and stubborn gardeners preserved these practices through generations of institutional neglect. This knowledge was preserved. University extension programs at Michigan State, Penn State, Cornell, and Ohio State published field research confirming what traditional farmers knew before the word microbiology existed. Meta-analyses published between 2021 and 2026 documented what no bag of synthetic fertilizer has ever replicated: that permanent productivity grows from a living soil population, not from soluble chemistry purchased each spring. Indigenous no-till systems in the Americas preserved mycorrhizal networks for centuries across corn, bean, and squash plantings. European peasant farmers rotated grain with legumes and cover crops for 1,000 years before synthetic nitrogen existed, building soils that fed the continent. Japanese natural farming traditions, preserved through the 20th century by figures like Masanobu Fukuoka, documented yields on untilled, mulched, diversely planted land that matched or exceeded industrial monocultures. They did not need a soil testing laboratory. They needed soil that worked on its own terms. Preserved soil traditions matter. Today, that knowledge is accelerating, not because the industry permitted it, because the research became undeniable, and because home growers, frustrated with expensive inputs that produced marginal results, started asking why the forest floor grows lush without a single bag of fertilizer. The silence around these practices is breaking. You can grow what they silenced. Start where you are. If your soil is biologically dead and compacted, begin with a heavy layer of compost and permanent mulch. If your beds are tilled every spring, stop tilling this fall and cover the surface instead. If your garden sits bare through the winter, plant a cover crop mix in September and watch what the following season produces. If you rotate nothing, begin with a simple 3-year rotation of legumes, fruiting crops, and roots across your beds. Start small and be consistent. The total investment for all 5 practices applied to a 1,000 square foot garden runs under $50 in seed and mulch material, less than most gardeners spend on synthetic inputs in a single month. These practices do not leave your soil depleted at the end of the season. They leave it measurably more alive than when you started. The mycorrhizal networks rebuild themselves. The bacterial populations compound. The earthworm counts climb year after year without a single additional purchase. The multi-billion-dollar agrochemical industry profits from a cycle of dependency that begins with the assumption that soil is dead. These 5 practices begin with the opposite truth, that soil is the most alive ecosystem on the planet, and when you feed it, protect it, and plant into it with diversity, it will feed you back beyond what any bag of synthetic nitrogen has ever promised. The knowledge was never lost. It was waiting in the rhizosphere, in the research literature, in the hands of farmers who never needed a soluble fertilizer to grow food. If this seed was finally freed for you, subscribe to Hank's gardening journey and hit the bell. Every like and every share helps break the silence. The next seed breaks free soon.